Welcome, grade 9 students of St. Paul American School Clark, to today's Explainer. Today, we are going to literally decode the blueprint of life itself. We're diving into Unit 3, Lesson 5, exploring DNA as the genetic material, along with RNA and proteins. It is a really fascinating journey into the microscopic world that makes you, well, you. So let's quickly go over our roadmap for today. We'll start with our lesson objectives, move into the history of DNA as the genetic material, dive into the central dogma, look at a real-world example of assembling a protein, do a quick misconception check, and finish up with a formative check and recap. All right, first up, our lesson objectives. By the time we wrap up this explainer, you're going to know exactly how early scientists discovered the genetic code. You'll be able to explain the physical structure of DNA, define what biologists call the central dogma, and detail the exact steps your cells take to translate genetic instructions into the proteins that physically build your body. Let's jump into Section 2, DNA as the genetic material. You know, it actually took over 30 years of incredibly careful, methodical experiments to finally confirm that DNA holds our genetic secrets. Back in the 1920s, Frederick Griffith noticed something strange. A harmless strain of bacteria could somehow be transformed into a deadly strain by picking up a mystery substance from dead bacteria. Fast forward to the 1940s, Oswald Avery and his team proved that when they selectively destroyed DNA, that transformation stopped cold. And finally, in the 1950s, Alfred Hershey and Martha Chase used viruses, which, by the way, are basically just DNA wrapped in a protein coat, to seal the deal. By tracking radioactive labels, they proved that viruses inject DNA, not protein, into bacteria to reproduce. So DNA was officially crowned as the genetic material. And thanks to brilliant scientists like Rosalind Franklin, James Watson, and Francis Crick, we know DNA forms an iconic double helix. Picture a spiraling staircase. The sides of the staircase are made of a sugar phosphate backbone, and the steps, those are made of nitrogen bases, and they follow really strict base pairing rules discovered by Erwin Chargaff. Adenine always pairs with thymine, and cytosine always pairs with guanine. So A bonds with T, C bonds with G. This perfect pairing is what keeps the distance between the two chains constant, maintaining that beautifully uniform spiral. If we zoom in really closely on that staircase, we see it's made of repeating units called nucleotides. A single nucleotide consists of 3 specific parts: a phosphate group, a nitrogen base, and the 5-carbon sugar. Now, in DNA, that sugar is called deoxyribose, and that is exactly what gives DNA, deoxyribonucleic acid, its famous name.
But here's the thing. DNA doesn't work alone. To understand how the cell actually functions, we have to contrast our heavy, double-stranded DNA, which stays locked securely inside the cell's nucleus, with its nimble messenger counterpart, RNA. RNA stands for ribonucleic acid. It's usually single-stranded, and it has a ribose sugar instead of deoxyribose. Crucially, though, RNA replaces the base thymine with a completely different base called uracil. So when dealing with RNA, adenine pairs with uracil, not thymine.
Moving right along to Section 3, the central dogma.
This right here is absolutely crucial. The central dogma of molecular biology states that genetic information flows in one direction only, from DNA to RNA to protein. Basically, DNA safely stores the instructions. RNA copies and carries those instructions, and proteins do the actual physical work, making up your tissues, enzymes, and all your cellular machinery. To make that leap from RNA to protein, we rely on 3 different types of RNA working together. Think of your cell as a factory. Messenger RNA, or mRNA, is the photocopy of the original DNA blueprint. It carries the code out of the nucleus and into the cytoplasm. Then you have ribosomal RNA, or rRNA, which actually makes up the ribosomes, the physical assembly line machinery of our factory. And finally, transfer RNA, or tRNA, acts like a fleet of tiny delivery trucks. Each tRNA carries one specific amino acid, the raw building blocks of proteins, and delivers it right to the ribosome assembly line.
Let's see this in action in Section 4, assembling a protein.
The entire process of building a protein from an mRNA template is called translation, and it happens in 3 very distinct phases. First up is initiation. The ribosome latches onto the mRNA transcript and scans it for a specific 3-letter code called a start codon, just so it knows exactly where to begin. Next is elongation. The ribosome reads the mRNA code in 3-letter chunks. With every chunk it reads, a tRNA delivery truck with the perfectly matching anticodon docks at the ribosome and drops off its specific amino acid. These amino acids link together one by one to form a growing chain. Finally, we hit termination. The ribosome encounters a stop codon on the mRNA. This is basically a red light. The factory machinery disassembles, and the newly built protein chain is released to fold into its final 3D shape and get to work in your body.
All right, Section 5, a quick misconception check.
Because we always hear people call DNA the blueprint of life, it is incredibly easy to assume this master molecule does all the heavy lifting itself. But is that actually true? Does DNA build proteins directly?
The answer is a resounding no. Think of DNA as the factory manager. The manager never, ever leaves the secure head office, which is the nucleus. DNA is just way too valuable to risk getting damaged out in the cytoplasm. Instead, it relies entirely on its RNA messengers to copy the instructions, leave the office, and get the actual building done on the factory floor by the ribosomes. Honestly, without RNA, your DNA is just a locked book of unread instructions.
Let's wrap things up with Section 6, formative check and recap.
Take just a quick mental second here. Try to remember the one-way flow of information we discussed, and recall Chargaff's rule for the nitrogen base adenosine. Got it?
Okay, if you said that the central dogma flows from DNA to RNA to protein, and that adenine always pairs with thymine in DNA, you have absolutely nailed the main takeaways of today's lesson. To quickly recap our journey, we saw how Griffith, Avery, and Hershey and Chase proved DNA is our genetic material. We explored how instructions flow through the central dogma. We reviewed the AT and CG base pairs of the double helix, and we broke down how translation turns an mRNA message into a functioning protein through initiation, elongation, and termination at the ribosome.
I'm going to leave you with this pretty mind-blowing fact straight from our sources. It takes an E. coli bacterium just 0.05 seconds to add a single amino acid during translation. Think about that. That means in the mere 10 seconds it takes to read a single sentence, one microscopic ribosome just flawlessly translated an entire 200 amino acid protein. It's almost unbelievable that this highly complex, high-speed factory machinery is operating continuously inside every single one of your cells every second of your life. Makes you wonder what other unseen miracles are happening inside you right now. Thanks for joining me on this Explainer Grade 9. Keep asking questions and keep exploring.